Hi everybody, this is our second molecular test for today, and we're going to test for an enzyme called enzyme M. If the plant has enzyme M, we're going to hopefully see that. So if you remember from earlier in the year, enzymes are proteins. And what makes proteins? Yeah, right? Proteins are made from your DNA. So if we can figure out if a protein or an enzyme specifically is present, then we will determine whether or not the DNA is similar. So it's kind of a way to tell us if there's similar DNA in the cell. So I have this indicator powder, and what we're looking for is some kind of chemical reaction. So I'm going to mix again the pigments with this indicator powder and in this well plate. And if there's a reaction, we will note that again on slide 15 in the column that says test for enzyme M. And you'll see I just have some paper chromatography drying here at the same time. So we're going to put a little indicator powder into this thing. It's called a well. Okay. Put some for BC, some for X, and I don't have to put a lot. Some for Y, and some for Z. So that's my indicator powder, right? Something I'm keeping the same in each of these situations here. Okay, cool. Now I'm going to put a couple drops of each kind of pigment, and we will focus on whether or not there is a reaction. Okay, so I've got some indicator powder in this well. I've got species B's pigment, and let's see what we got here. Oh, shoot. It's bubbling like nobody's business. So I would think when you mix the indicator powder and species Botanicurus, do you get a chemical reaction? Do you get some kind of fizzing? So write down whether there's fizzing or no fizzing for Botanicurus. All right, now let's go on to species X. Again, we're looking for which one is most similar to Botanicurus because it can make curol. So here we go. I don't really see any fizzing like last time. Kind of just sat there, right? There was no pssing kind of thing. So I definitely think there was no chemical reaction there, right? You can see no chemical reaction, just kind of sitting there on the surface. Okay, let's go on to species Y. Okay, we have our indicator powder. Here we go. Look, it's such a pretty, oh, dang, that bad boy is bubbling. So do you see a chemical reaction? And if there's a chemical reaction, that means that enzyme M is present. So when you see a chemical reaction, that fizzing, right, that chemical reaction means that enzyme M is present. Okay, and our last one, species Z. Okay, here we go. Oh, dang. Again, fizzing means we're doing a chemical reaction, and that fizzing is caused by enzyme M being present in that pigment. That's a pretty cool picture. Cool, so you will record all your data, right? BC, X, and Y, and Z. If there was fizzing, that means there was a chemical reaction present, and that means that enzyme M was present. Great job.